We will conclude the process of DNA replication in this video. I hope you have seen the part 1 of the process of DNA replication. So what we have learned so far is something like this. So we have a DNA that is to be replicated. The two strands of the DNA are anti-parallel, we know this, and they are held together by complementary base pairing, hydrogen bonds between complementary base pairing. And because DNA has been coiled molecule, and therefore it was very important to uncoil the DNA and start separating the two strands of the DNA from one another so that both the strands can act as template. And I told you in the last video that this thing would be done by an enzyme which is called as helicase, and this helicase number 1 did what is called as the unwinding of the coiled DNA and made a cut at ori. So we have discussed the significance of origin of replication site, and some of the hydrogen bonds have been cut, and this leads to the formation of what is called as a replication fork. So we have done this, that this leads to the formation of a replication fork. This will be a pretty long DNA molecule, and in this part you can see that the double-stranded DNA, the two constituent strands have been partly separated from each other, and this is where this inverted Y-shaped structure, this would be called as a replication fork. Then we said that to prevent the individual winding of the separated strand, and because the two strands are still complementary to each other, there is a danger that they may get re-annealed or rejoin with each other. So to prevent that, we used what are called as single strand binding proteins, and these single strand binding proteins, they separated these partially separated single strands so that they do not undergo coiling again or re-anneal. And then there was something happening, that was that the tension was developing ahead of the replication fork, and that was actually opposing the further opening of the helix. And for that, the tension developing ahead of the replication fork was reduced with the help of an enzyme which is called as topoisomerase that made a single stranded cut in DNA. And I told you that such an enzyme in bacteria would actually be called as a DNA gyrase. So now the thing is that this is the DNA. So we are talking of the formation of replication fork. The two strands are anti-parallel, and now we can start replicating DNA. That means we can start synthesis of DNA. Again and again, let me remind you that both strands will have to act as the template. You know about semi-conservative mode of DNA replication. Now the stage is set for this particular enzyme. We are saying DNA polymerase. So this enzyme, I told you actually it should be called as DNA dependent or DNA directed DNA polymerase. And I actually advised you to remember at least 2 of them in a prokaryotic cell. That would be DNA polymerase 1 and DNA polymerase 3. So let us recap a little bit of their functions. So DNA polymerase 3 and DNA polymerase 1 both are capable of synthesizing or polymerizing DNA or adding deoxyribonucleotides. So that means, but they do this only in 5' to 3' direction. So both the enzymes have a 5' to 3' synthesis activity. This is what we know. Number 2, I told you that there is an exonuclease activity as well. Exonuclease activity will be able to remove nucleotides from the end of the strand, not in the middle. So if it is not from the end, if it is somewhere in the middle or somewhere that is away from the ends of the nucleic acid strands, then the enzyme would not be called as an exonuclease, it will be called as an endonuclease. We will talk about a very important enzyme, restriction endonuclease, in your chapters on biotechnology. But let us understand that they both act as an exonuclease, which means that if these are the deoxyribonucleotides that have been joined to form a strand, let us say this is 3' direction, okay, let me make it other way around, this is 5' and this is 3' direction, then both enzymes have a 3' to 5' exonuclease activity. So this also has a 3' to 5' exonuclease activity. That means from this end they can remove nucleotide one by one. So that would be an exonuclease, and I told you specifically that this is going to be used now in proofreading. And one more very important thing is that because normally DNA polymerase 3 will be recruited at the replication fork, and therefore this is the enzyme that will be actually doing the exonuclease or proofreading function as well, although both enzymes are capable of doing it. Then I said that DNA polymerase 1 has a unique property. It has 5' to 3' exonuclease activity as well. That means it can remove the nucleotides from this end of the strand as well. This property is not present in DNA polymerase 3. So we have recapped what basically the DNA polymerases of a prokaryotic cell, especially 1 and 3, are capable of. Now let us come to a very important point, very important point. One is that DNA dependent DNA polymerase cannot initiate the synthesis. So that means it cannot add the initial nucleotides. It is not capable of initiating the synthesis. It requires a sort of preformed strand that can be actually extended by the DNA dependent DNA polymerase, which means that if this is the strand that is being replicated, the initiation part cannot be done by the DNA dependent DNA polymerase. It will not be able to start the process. But if it is provided with a small strand which has a 3' hydroxyl that is free to undergo bonding at this particular end. Remember, this is 3' to 5' DNA template. So the new strand is being described as being synthesized in 5' to 3' direction. What I am saying is that DNA polymerase will not be able to initiate the synthesis. It requires a 3' hydroxyl so that it can add deoxyribonucleotides to this particular end. So therefore it is imperative that for the action of DNA polymerase there must be a pre-existing short strand that can actually be elongated by DNA dependent DNA polymerase. I hope this thing is very clear to you. So let us repeat it again. We are saying that these enzymes they cannot initiate the process. That means the initial deoxyribonucleotides cannot be joined by them. They require at least a small or a short strand. Now this strand would be in 5' to 3' direction and would have a 3' hydroxyl that is free for bonding. This is where the deoxyribonucleotides can be joined. In last video I told you that this is the specific point by which there is a directional sensitivity of DNA polymerase. That means they can synthesize only in 5' to 3' direction. Now this poses a very unique problem in DNA replication, because the two strands of the DNA that has to be replicated, both the strands are to be replicated. One has opened in 3' to 5' direction, the other has opened in 5' to 3' direction. So there would be some complexity here. So again, please remember, cannot initiate but can extend any strand. This is a very important property. Number 2, they can synthesize only in 5' to 3' direction. Repeatedly I have told you why is it so. So I hope this thing is absolutely clear. Now let us see how these problems are overcome. So one thing is absolutely sure. If you want to synthesize DNA on the template of DNA, you will require DNA polymerase. And because DNA polymerase cannot begin the synthesis, then therefore something else must be synthesized in the beginning, and this thing cannot be DNA. Because again I am saying that if I am making any DNA, it has to be done by DNA polymerase, and it does not have the capability of initiating the synthesis. So what happens? So instead a very special enzyme which is called as a primase. So this primase is actually an RNA polymerase. Now if DNA polymerase makes DNA, then RNA polymerase will synthesize a strand of ribonucleic acid or RNA. So this primase is a very special RNA polymerase, that is DNA dependent RNA polymerase. That means it can synthesize RNA on the template of DNA, and it does so in 5' to 3' direction. Now this is very, very critical to understand. So we are first actually synthesizing not DNA, but we are synthesizing what is called as a small or a short segment of RNA nucleotides. And this RNA nucleotide short segment is appropriately called as a primer. And this primer is made up of ribonucleotides. And please remember that the enzyme is actually called as a primase. So this is how this thing is sorted out. It cannot initiate, no problem. So what we make a segment of actually RNA first, and this is the short segment that will actually be extended by the DNA polymerase, because now the requirement of 3' hydroxyl for bonding is being fulfilled, and the directional sensitivity of synthesis of DNA polymerase can be maintained. So this is one thing. So that means the problem of initiation can actually be taken care of by synthesis of a primer. Now this is a very, very important point. That means the RNA polymerase doesn't have any problem in initiating this. So this is one more point that we can say in the favor of that, this being an RNA word. So the first genetic molecule or the first genetic informational molecule of the life on this earth may well have been a ribonucleic acid and not DNA. I have discussed this very important concept in a separate video, that DNA may have actually evolved from structural modifications in RNA, and because DNA is more stable than RNA, the function of storage of genetic information goes to DNA, and the flow or the transmission of the genetic information in a living cell, this function was primarily taken over by RNA. So that means this is very, very clear. So in the beginning, actually before the DNA polymerase is able to work, RNA primer has been synthesized. Now this is very, very simple. I told you in one more video that we will use these things, what is called as ribonucleoside triphosphates. So I am not going into details again. I am saying that the substrate for DNA dependent DNA polymerase would be deoxyribonucleoside triphosphate. This is very similar to ATP, the energy currency which is used for providing the physiological processes in the body, or it is also providing energy for what is called as the endothermic reactions that are taking part in the body. So please see that video, what exactly is a ribonucleoside triphosphate. That will, number one, act as a substrate for DNA dependent DNA polymerase, and number two, will also be the source of energy, because it will also have two terminal bonds between phosphate groups as high energy bonds. That means breaking that bonds will give you a large amount of energy that will be required for polymerization. Now the thing is very simple on this particular strand, the strand that has been opened in 3' to 5' direction. So we are talking of one of these two strands. We are talking of the strand that has opened in 3' to 5' direction. We have synthesized the primer, and now the DNA dependent DNA polymerase takes over, and based on the complementary base pairing, it will attach deoxyribonucleotides using deoxyribonucleoside triphosphates as substrate as well as source of energy to the 3' end of the strand. So that is, and therefore it is maintaining its direction, which is 5' to 3'. So this is what is called as elongation of the primer, and actually it is more appropriately called as an extension of the primer. So this is the function that is actually being done by DNA polymerase, that it extends the primer. Now please remember the things that I have made in red color, they are now deoxyribonucleotides. This is DNA molecule that is being synthesized, but what is being extended is an RNA molecule, which is primer. So I hope this thing is absolutely clear. Now let us take care of the other strand. Now it is pretty evident that the direction of DNA synthesis cannot be like this, because if I am synthesizing any strand on this particular template, then the direction would become 3' to 5', and that is not the capability of DNA polymerase. So that means something else has to happen here. So this thing cannot happen in the same direction. It cannot happen in the same direction. So two molecules of DNA polymerases will be recruited at the replication fork. Things are very complex. One of the strand actually loops over, and both the DNA polymerases actually move towards the same direction. But actually it is very important to understand that we are making a functional diagram of DNA replication, simplifying the process. This strand has been opened in 5' to 3' direction. It is not possible to synthesize a DNA strand on this molecule in this direction, because that would be 3' to 5'. So what exactly is done? So what exactly is done, that a primer is synthesized here. So that means maintaining the directional sensitivity. Now you will allow this particular strand to open up a little bit, and then you will synthesize a primer. Remember, if I am synthesizing the primer in this direction, this direction is 5' to 3'. This is the 5' end, and this is the 3' end of the primer. And again, because I am doing it in the reverse manner, now this is very easy to understand that again at the end of the primer, the 3' hydroxyl group will be available for the formation of phosphodiester bond, and this primer can now be elongated or extended by the DNA dependent DNA polymerase by adding deoxyribonucleotides in 5' to 3' direction, and it is again based on complementary base pairing. So that means the two problems have been taken care of. So this is very, very important to understand. So this is what happens on the two strands. Remember, both strands are acting actually as template. Now let us see what happens further down the road. So what happens further? What happens is that the helix opens up more. So that means the enzymes, the helix has been opened up more, and let us say something like this has happened. It has opened in a larger length, and this was the original strand 3' to 5'. This was 5' to 3', and if you remember that we had synthesized a primer here in 5' direction to 3' direction and extended the primer by adding the deoxyribonucleotides. And this strand will keep on opening, and the DNA polymerase that is recruited on this particular strand based on complementary base pairing will keep on adding deoxyribonucleotides in a continuous manner. Mind my words. So that means this is how it is happening. And if you remember, on the other strand we had initially synthesized primer in keeping the direction which is 5' to 3'. This is what happened on the other strand, and we extended this particular primer in a short distance. On this particular strand we are still keeping the directional sensitivity of the DNA polymerase, and this is the DNA segment. But when it opens up further, again on this strand you will again have to synthesize a primer segment. So this segment, this primer segment now is not required on this particular strand that is being continuously synthesized. But the problem is on the other strand, we have again synthesized a primer and then again extended the primer, keeping the directional sensitivity of DNA dependent DNA polymerase. I hope this thing is absolutely clear. Now please see that one of these strands is being continuously replicated, while the other strand is discontinuously replicated in short fragments. Each fragment is having in the initial part an RNA primer. This is very, very important to understand. So we are saying that this particular strand has been synthesized continuously, and it is continuously means that you are requiring only one primer and you keep on extending the primer as and when the helix gets opened towards the replication fork. So this is being replicated continuously. The second term I am using is that it is being replicated towards the replication fork. So this is happening towards the replication fork. You can see it is moving towards the replication fork. What about the other strand? The other strand is being replicated, we will say discontinuously. So discontinuously, and it will be away from replication fork. And in these fragments, the fragments of the discontinuously synthesized DNA strand, these fragments are called as Okazaki fragments. And remember that each Okazaki fragment in the beginning has a small part of or small stretch of RNA molecule which is called as the primer. So I hope this is absolutely clear, because one of the strand is being synthesized continuously, the other is discontinuously. The one which is synthesized continuously, this is strand. I am talking about this strand that is being synthesized continuously is appropriately called as the leading strand. So this strand is called as the leading strand, while the discontinuously synthesized strand is called as a lagging strand. So one of the strands is leading, the other is lagging, and because one is continuous, the other is discontinuous. So if you have read your NCERT well, it also says that DNA replication is not only semi-conservative but is semi-discontinuous as well. So it is semi-discontinuous as well. So now this is appropriate. Now you can understand this will keep on opening, and what is going to happen, this will be synthesized in a continuous manner and small Okazaki fragments will be formed on the lagging strand. So now there is an important thing to consider here. What happens if there is a mistake by DNA polymerase? I told you this mistake can be in 1 in 10 to the power 6 or 10 to the power 7. It has a very, very high fidelity rate, but it can commit a mistake in adding deoxyribonucleotides, and you can understand the magnitude that I am talking about, 1 in 10 to the power 6 or 10 to the power 7. So let us say that a wrong base or a wrong nucleotide has been inserted by the DNA polymerase. So this should not have been there. This is not following, say for example, the complementary base pairing, and a mistake has been committed by the DNA dependent DNA polymerase. Now because we know that this DNA polymerase also has a 3' to 5' exonuclease activity, and please remember that this has been added to the 3' end of the synthesized strand. So the strand that is being elongated, so this particular thing, this is a wrong base that has been inserted at the 3' end of the elongating strand. Now because it has 3' to 5' exonuclease activity, so this can actually be removed. So that means that the DNA polymerase is very capable of recognizing the mistake, and if it does so, then it is able to remove this and replace it with the correct nucleotide. So that means this has been removed. This is now possible because I am removing it from the end, and that is what is an exonuclease. And because I am removing it from the 3' end, so it requires a 3' to 5' exonuclease activity that is actually present in DNA polymerase 3, which will be the main recruiting enzyme that will actually be synthesizing the DNA in most of the cases. But remember, DNA polymerase 1 also has this capability. So this removal, identification of the wrong insertion of the base, removal by exonuclease activity, and replacement by the correct nucleotide, this thing is called as proofreading function. And this proofreading function actually gives a very high fidelity to DNA polymerase, and therefore the error in DNA replication is of a very low magnitude, 1 in 10 to the power 6 and 1 in 10 to the power 7. We have repeatedly told you. So this is what is called as the proofreading function of DNA polymerase. Now let us see what happens when everything goes well. Then let me rub all these things, and let us see that, okay, our DNA molecule has been now entirely separated, the two strands. One of the strand originally was 3' to 5', the other was anti-parallel 5' to 3'. We synthesized an RNA primer here. This is the 5' direction. So we are synthesizing or we are replicating this strand in 5' to 3' direction. That is the direction of the DNA polymerase. And now the DNA polymerase extends this particular strand continuously. This is the leading strand. It just reads the bases that are present on the parent or the template strand and add complementary bases and elongating the strand. Remember, proofreading has been done, and let us say this thing has been finished. So this thing has been finished. Now what happens to the other strand? We know that this was the lagging strand and it was actually synthesized discontinuously, and there were Okazaki fragments that were formed, and they were formed away from the replication, and short stretches were actually replicated. And you can see this is the formation of Okazaki fragments, the discontinuous mode of replication of what is called as the lagging strand away from the replication fork. And you can see I have made a primer for each Okazaki fragment. So I hope this thing is absolutely clear. This thing is absolutely clear. So this has been synthesized or replicated discontinuously, and this has been replicated continuously, making it a semi-discontinuous mode of DNA replication. Now the part is that now these primers must be removed, and the gaps should be filled by deoxyribonucleotides. So now it is very important that these primers should be removed, and these primers can be removed with the help of DNA polymerase 1 only. So this is the only enzyme that is capable of removal of primers, for the simple reason that this enzyme also has what is called as 5' to 3' exonuclease activity. So it will remove the primers. So the primers, remember, have been synthesized in 5' to 3' direction, and therefore you require a 5' to 3' exonuclease activity, and that exonuclease activity is present in DNA polymerase 1, and that actually removes the primers and actually fills the gap with deoxyribonucleotides, because now there is already a strand that has been synthesized, so there is no point of initiation. So you can say that what has happened, that these have been removed and replaced. So this is very, very simple. So this thing is done by DNA polymerase 1. Very, very important role, removal of primer, 5' to 3' exonuclease activity. Now on the lagging strand, one function is still left. The idea is now to make phosphodiester bond and join the Okazaki fragments. Remember, this will not be required on the leading strand. So the action that I am talking about is limited only to the lagging strand, which has been synthesized discontinuously. On this strand, there has to be the joining of the Okazaki fragments, and this enzyme would be DNA ligase. So DNA ligase on the lagging strand will actually join the Okazaki fragments, completing the process of DNA replication. I hope you have understood the basic steps of DNA replication. This is very, very important. Let me remind you and just recap very fast that the first enzyme to act was helicase. Unwinding, cut at ori, partly separating the two strands of the double helix, leading to the formation of replication fork. This was step number 1. Then stabilizing of the partially separated strands by single strand binding proteins. Then the tension that was developing ahead of the replication fork was reduced with the help of topoisomerase or DNA gyrase. This thing is absolutely clear. Then comes the function of DNA dependent DNA polymerases. They cannot initiate. They have a directional sensitivity which is 5' to 3' only. So one of the strand. So therefore, because they cannot initiate, you synthesized a primer, and that was done by a very special enzyme which is called as primase. So this is a short stretch of RNA molecule that has been synthesized first, and this will be elongated by DNA dependent DNA polymerase based on complementary base pairing. One of the strands would be replicated continuously towards the replication fork and would be called as the leading strand. The other strand would be replicated discontinuously away from the replication fork and would be called as the lagging strand. And if required, there would be proofreading because of 3' to 5' exonuclease activity. Then finally you will have to remove the primers. So the primers can actually be removed only by DNA dependent DNA polymerase 1 because it has a 5' to 3' exonuclease activity. The last thing to be done would be on the lagging strand, where you join together the Okazaki fragments, and this would be possible with the help of again a very, very important enzyme which is called as DNA ligase. So I hope the process is absolutely clear. Now we have two daughter strands of DNA molecules which are actually genetically identical. The DNA has replicated itself. Every time a cell divides, before division the DNA has to replicate, or we should say that if a cell actually replicates its DNA, then it is committed to divide. Otherwise there is no point of replicating the DNA. We know that DNA replication will be able to maintain the continuity between the populations of the cell. So that means not only a single zygote can become a body of more than 50 trillion cells as in human beings. You can imagine the amount of cell division that has taken place, and therefore the amount of DNA replication that has taken place. It will also lead to the formation of gametes. We have discussed this thing in mitosis and meiosis, cell cycle and cell division, some very, very important concepts about DNA replication. And please remember from your class 11 syllabus that in the cell cycle, after the G1 phase, or I should say between G1 and G2 phase, there was a phase which is called as S phase, and that S phase, S means synthesis. This is where DNA replication or DNA synthesis actually occurs. So this is for eukaryotic cells. So this is what happens in the cell cycle of a eukaryotic cell. A very important thing is because the DNA of prokaryotes is actually circular. I told you that we have understood the functional aspects of the DNA replication by simplifying the process. So what we are saying here is that because it is a circular double-stranded DNA, so there would be a single ori. there would be a single ori. So we are talking of prokaryotes. There would be a single ori, and if I cut the hydrogen bonds here, you can very well imagine that it will lead to formation of something like this. So you can see that the replication fork actually moves bidirectionally. So that means there is a single ori in prokaryotes and replication forks move bidirectionally. So somewhere when you look at the middle of the process, when the process has undergone a considerable amount, and middle of the process, this is how it will be replicated, and this resembles the Greek letter theta and sometimes is called as theta replication. So I am talking of prokaryotes. There is a single ori and the replication fork moves bidirectionally. The DNA is very long. So DNA is very long in eukaryotes, and therefore a single ori will not suffice, and therefore there would be multiple oris. So there are multiple ori in eukaryotic DNA, and again the replication fork will move bidirectionally. That means if you are somehow able to see a living cell replicating its DNA, then you will see that there are some bubbles that are being created. So here again you can see a cut has been made, and therefore you can see something like this will happen, what is called as a bubble. So you can see replicating DNA having some bubbles. You know that this is where the replication has actually started. And please understand that here as well the replication fork moves bidirectionally. So please understand that the replication fork moves bidirectionally, because we have simplified the diagram. We started from one end. It does not mean that in eukaryotes the replication fork actually moves unidirectionally. It actually moves bidirectionally as well. So if I compare this thing with this particular phenomena, I will say that there are multiple ori in eukaryotes, and the replication fork actually again moves bidirectionally. So this is very important to understand that this is a very important difference between prokaryotes and eukaryotes. In the chapter in common human diseases, there would be a subtopic which is called as cancer, and there the ends of the chromosome are actually called as telomeres, because eukaryotic DNA is a linear DNA. It is not possible to replicate the DNA to the very ends. So it is not possible to replicate the DNA to the very ends, and therefore each time the DNA replicates or cell divides, the length of the telomeres gets shortened, and if it reaches a particular critical limit, below that the cell actually does not divide, what is called as Hayflick limit. And there is a very important enzyme that we have discussed elsewhere as well, telomerase, that would be very important in the pathogenesis of cancer. So I hope you have understood this very important concept in molecular biology, the DNA replication. Before saying thank you to you, let me remind you the sequence of the enzymes once again. Helicase, then I said single strand binding proteins, then I said topoisomerase, then the DNA polymerases. That means there was a primer. That primer, actually there was RNA primer or primase enzyme which was extended by the DNA polymerase. Remember the proofreading function, and then removal of primer by polymerase 1, and finally the ligation of the Okazaki fragments on the lagging strand that was done by DNA polymerase, that was done by DNA ligase. I hope you will remember the sequence of the enzymes. A question in NEET exam is virtually a guarantee from this particular topic. Thank you so much.